Today we are going to discuss answer scheme for experiment 6, isolating DNA. In your lab report, make sure that you write all content below: title, objective, procedure, in flow chart, diagram or you can draw, result and observation, discussion, question and answer, conclusion, and also references. First, you need to write a title. Okay, so title for today's experiment is isolating DNA. The second one is objective. We have only one objective for this experiment, to isolate DNA from plant tissue. And the third one is a method. You can summarize the procedure of this experiment using flow chart or using suitable label diagram. You also can draw your procedure. Let us discuss result and observation of our experiment 6. Firstly, you have to make sure that you draw 3 layers of solution. The first solution, the second layer solution, and the last one solution. Then you need to draw the first solution with alcohol, the second layer of solution with the DNA filament in supernatant layer, and the last one at the bottom layer you have to label with the sieve liquid. So the total up for this result or observation is 5 marks. The next one we will discuss about discussion. In this experiment, you have to discuss the observation of extract plant's DNA in terms of color and texture, and also its location in container. So the result of observation is: first, DNA is white and transparent in color, 1 marks. It is a jelly-like mucus filament structure, 1. And it can be found in the middle layer of alcohol, which is located at the top layer, and liquid at the bottom layer, the sieve liquid at the bottom layer. Okay, so we got all together 3 marks for first discussion. For the second discussion, we have to discuss about the 3 precaution steps. The first step is alcohol must be chilled before usage to ensure that DNA well precipitated, 1 mark. The second one is avoid formation of foam by stirring gently, 1 mark. And the last precaution step is ice chill alcohol is poured at the flat angle to ensure the formation of separated layer, 1 mark. So the total marks for the second discussion is 3 marks. Next, we will discuss about the question and answer. Write the significance of this step. The first step, mechanical disruption using mortar and pestle to break down open the cell. The second one is tissue will break into the small pieces, and mechanical disruption is particular importance when using plant cell because it have a tough cell wall. So the significance for this step is 1 marks. Second, peel, slice and mash the fruits. So the significance for this step is to lyase or break down the fruit cell wall, 1 marks. The third significant step is apply the flat angle technique. Why we need to apply the flat angle technique because we want to make sure that both liquid do not mix. Or you also can write alcohol will form a separate layer on top of the sieve liquid. Also 1 marks. The second question is about the purpose of addition of salt detergent solution. So salt is used to make sure the positive charge of sodium ion in salt help the negatively charged phosphate group that run along the backbone of DNA. The second one, salt also neutralize DNA charge. Salt make DNA less hydrophilic. Salt make DNA become less soluble in water. And salt also help to remove protein that bounded to DNA. So you have to choose any one answer. This is equal to 1 marks. The purpose of detergent solution is detergent can break down the lipid of cell membrane and nuclei. Also, DNA is released as these membranes are destructed. Detergent also used to emulsify fat, mostly phospholipid bilayer. Detergent also used to create a polar environment to dissolve DNA. And the last one, detergent used to pulling apart the membrane fats and protein to release DNA from cell. You can choose any two answer. So this is equal to 2 marks. The next question is the addition of ice-chill alcohol. According to this step, ice-chill alcohol is used to make the DNA is soluble in water but insoluble in the presence of salt and alcohol. Alcohol also very useful in preserving DNA. It is become visible and easily to precipitate. So total 3 marks for this question. Okay, why we need to put the solution into 60 degree of water? Okay, because warm water is used to denature protein and enzyme DNAs that can degrade DNA. Also to soften the tissue and allow the solution to penetrate into cell. So total marks for this question is 2. The next question is state 3 organelles in plant cell which contain DNA. The first one is nucleus, second one is mitochondrion, and the last one is chloroplast. Total marks for this is R3. The next question is extraction of DNA in plant cell nucleus differ from animal cell. Briefly explain. DNA from plant cell are more difficult to extract due to the presence of the cell wall. In contrast, the animal cell does not have cell wall. So if you write about both to plant and animal cell, you will get 2 marks. Okay, and the last one, what is the functions of DNA? So we need to write function of DNA is to carry genetic information for the development and function of living things. Equal to 1 mark. We're done for the question and answer. So we need to write the conclusion for this experiment where you have to answer the objective given. For the conclusion, you have to write about the plant DNA was isolated using this technique. and it can be seen as white precipitate between alcohol and sieve liquid. If you write complete conclusion, you are given 2 marks. And don't forget to write references based on APA. An example of one reference given here is a Campbell, Urey, Chain, Westermann, Minor Sky, Rees, 2017, Biology: A Global Approach, page 367 to 372, New York, Pearson Education. Even we have no marks for this reference, but you have to write it. Okay, so best luck for your practical test. And don't forget the important dates we have on Monday, 27th of September. We have a practical test start. Tuesday, 28th of September, you need to submit your lab report. And on Friday, 1st of October, you have to submit your video. That's all. Thank you.